Okay. Hi there, Epidemics. Hope everybody's doing well again today. Our usual reminders, assignments are due tomorrow before midnight. New assignments will go up either tomorrow or Saturday. And the project, of course, is due 1 month from today. So last time we were kind of talking about how society was behaving and reacting to the second plague pandemic. And so now we're going to start talking about the decline of the second plague pandemic. So I want to remind you of our dates here. The second plague pandemic began in Europe and the Middle East in 1348. It was probably started before that in parts of Asia, but again, I just don't have good information for what was going on there. The second plague pandemic did not end until 1840. So that's almost 500 years people were dealing with this horrific, highly deadly, highly contagious disease. So let's talk about the decline and end of this plague pandemic. So it was kind of gradual. After the Black Death, those first 5 years, we had continual outbreaks. And the outbreaks started to decline in frequency, so they weren't coming as regularly. They were less severe. We think that the severity went down for a couple of different reasons. One of which was immunity in people. So this arose because those who survived had genetic advantages. Something in their genes gave them an advantage. They made it more likely for them to survive the plague. Those who were highly vulnerable did not have the opportunity to reproduce. They didn't survive. So we think the immunity in the population, not an individual's immunity, but the population immunity went up. We also saw changes in the rats. So when we talked about the rats at the beginning, the rats that were spreading the plague, living close to people, their fleas would go from the rat to the people because they were in close contact. The black rat was replaced by the brown rat. So the brown rat's a different species. It is a larger rat. It is more aggressive. So the black rat was, semi-domesticated. It liked people. People liked the rat. It wasn't very aggressive. It was very comfortable hanging around people. They could be tamed. They could be trained. Sometimes they were kept as pets, sort of pets. But the brown rat wouldn't tolerate this. The brown rat did not like people, even though it had an advantage living around people and around populated areas. It did well there. It was more aggressive to people. It would avoid people on a personal basis. And it was therefore less efficient at spreading the plague because they weren't living in the walls of houses. So this change in rats, the black rat, which was excellent at spreading plague to people, being replaced by the brown rat, who wasn't good at this, meant people were being exposed to plague infected fleas less frequently. We see improvements in nutrition. Poor nutrition results in poor immune response. And thus poor immunity could be passed. So if a pregnant woman was malnourished, her child would be born with a weaker immune system. And you never recover from that. Quarantines, we talked last time about how they don't work that well, but they can. They do reduce the exposure to the plague, especially in port cities where you could have people quarantining on an island. When we look at the bacteria, it also evolved during this time period. When we look at the bacteria from different places around the world, from the time period of the Black Death, and yes, we can get the DNA from the bacteria from people who died during the Black Death. This doesn't mean it's contagious bacteria. It means we can get the DNA. And so we see that there is a increase over time in genetic diversity of the bacteria, which is a good sign that it's evolving. And more importantly, we see loss of two different virulence-related genes. Remember, virulence genes, virulence factors are what increases the ability of the bacteria to spread and increases the severity of the infection. So because over time the bacteria lost these genes, the case fatality rate declined. So it wasn't as deadly towards the end of the second plague pandemic as it was at the beginning. We can also look at climate change. There was depopulation in Europe, which resulted in fields going fallow. So nobody was maintaining the farmland. And so a lot of the farmland turned back into forests. It's questionable whether this caused climate change or not, but it could very well have contributed to it. And the European winters got harsher. Harsh winter, fewer rats surviving, fewer fleas surviving. To give you an idea, at the beginning of the Black Death, remember we talked about going through, just before the Black Death, there was a warm period in Europe. And in London, they were able to grow orange trees in London. Later on, during the second plague pandemic, London started having winter festivals on the Thames. So the main river that flows through London froze over to such a depth that people could walk, build temporary buildings, and have all kinds of things going on the river itself. We see changes in housing. My goodness, there were a lot of changes, but we're talking over, almost 500 years here. So housing changes. Mud floors were replaced with concrete. So that meant that rodents would have problems burrowing in from underground, for example. And it was easier to keep clean. Thatched roofs replaced with tile. While a lot of people think thatched roofs are cute, they're aesthetic, they're certainly very easy to build. but they also provide home for rats, fleas, all kinds of things can live in a thatched roof. When you have a tile roof, there's separation between the outside world and the inside world. You've got a better barrier there. Grain storage moved out of the house. So it used to be that people would store grain inside their houses, which of course attracts rats and blah, blah, blah. Move it out. Obviously, less attraction for the rats. We developed bar soap and increased bathing and washing. So we think of the Middle Ages, we think of the people there, thanks to pop culture, we think of them as being very dirty people. Well, they were by modern standards, but they did try. They would wash their face and their hands, but a full bath would be difficult. As we move forward in time, we start getting plumbing, we start getting bathhouses with heated water, so you could go there and take a bath. So we end up increasing the amount of bathing, increasing the amount of washing. And as the economic status of the individual went up, people were able to afford more clothes. So you didn't have one outfit that, you know, washing it meant you had to wear wet clothes. You would have multiple outfits and you could clean them more frequently. So this is where we stand as we move forward through the second plague pandemic. We've got all of these changes going on, all of these things that are making it harder for the plague to infect people. And then in September 1666, the Great Fire of London happened. So London has, for most of the time period we're talking about and all the way into today, it's one of the world's biggest cities. In the 1660s, it was, you know, it was already one of the most modern, one of the biggest cities in the world. The Great Fire in London swept through in 1666. It destroyed a huge portion of the city. Buildings burned. And this, while a horrible thing, you know, people lost their lives, they lost their houses, they, you know, all kinds of stuff. It allowed for rebuilding. And so the rebuilding after the Great Fire of London was with what was then considered modern construction. Those things I was talking about: concrete floors, tile roofs, rat-resistant materials. And the fire also killed a lot of the rats and the fleas. So the rat population went down. Before the Great Fire of London, London was having an outbreak of the plague. This fire stopped that outbreak cold. William, yes, go ahead.
Isn't the Great Fire of London what inspired the nursery rhyme about the London Bridge?
It's possible. I don't know the correct answer to that. It makes sense. This picture here shows the old London Bridge, and you'll notice it had houses on it. It was very different than the modern London Bridge. So it would make sense that it would have been destroyed at this time, but I don't know about the nursery rhyme. It's an interesting question. So on the plus side, people around the world said, oh, look, if we rebuild with modern construction, we can, reduce the risk of plague. Unfortunately, what it ended up was being the inspiration of burning tenements, so poor communities. So, of course, the rich were not, burning their buildings down, but they would burn down poor communities in an attempt to do the same thing that happened in London, deal with the rat population. And one of the cities where this happened was Calcutta, where people were literally evicted from their homes. The whole area was burned to the ground. It was actually socially a really, really nasty thing. So those are the kinds of things that contributed to the end of the second plague pandemic. It kind of just, petered out. Didn't go out with a bang, went out with a whimper. And that lasted for just a few years. And then we have the third plague pandemic. So I'm going to talk a little bit about this, but not a lot. The third plague pandemic ran for about 100 years. It ended in 1960. Your parents were alive while the third plague pandemic was going on. Probably. Hang on. Well, at least your grandparents were. Depends on how young you are. The third plague pandemic started in Yunnan, China. And there appear to be two different plague pandemics that started about the same time. They were genetically different. One of them was the bubonic plague, kind of like what we saw in the second plague pandemic. This, of course, remember, is the less deadly form. And the other one was predominantly pneumatic. So it spread through respiration. And that one, if you recall, was one of the deadlier ones. This third plague pandemic predominantly impacted China and India, and it killed more than 12 million people, mostly in India. But we had outbreaks around the world. If we look at our little map here, this is where the Yunnan province in China is. So we think this is the start of that outbreak. And it spread to, I think that's Kazakhstan. It spread to Japan, spread to India, Africa, Australia, Hawaii, and the San Francisco area in the US. San Francisco got hit about 1900. A second, the genetically different version of the plague, I don't know if it jumped from rats into humans or if it switched, if there was a genetic change from bubonic to pneumonic in India, but that one started in India, spread to Europe, the Middle East, Madagascar, parts of Africa, South America, and then to South Africa. So again, this was a large percentage of the world being impacted. The outbreak in San Francisco started in the early 1900s. The government of the US, actually, I don't know if California was a state at that point. Anyway, the local San Francisco government denied that plague was happening there. They denied it because of economic concerns. Once again, the economy was being more important than people. we think that this second plague pandemic was related to the Panthay Rebellion. So the Panthay Rebellion was related to racial tensions and led to multi-ethnic insurgencies, multi-ethnic fights against the government. So there was a government that was predominantly one ethnicity. They weren't being fair to the other ethnicities in the community, and there was a rebellion. And we think that troops began moving and refugees began moving what could have been just a local outbreak to other regions. So because India was highly impacted, let's talk a little tiny bit about India at this point. During this third plague pandemic, India was ruled by the colonial government, which was established by the British. So the British had invaded and conquered India and installed Brits as the new government. So you can see all of these very wealthy looking Indian men, and this guy's the boss, the Brit. So the colonial government did the kinds of things that they'd seen work before. They enforced quarantines, isolated infected people, restricted travel. And one of the things they did, which was probably not great, was they excluded traditional medical practices. So the British felt that their medical practices were clearly superior to what the people in India were doing. So you got to do the British way. There were special plague committees that were supported by British military. And these repressive actions during this third plague pandemic, and the fact that it didn't really work that well, contributed to the early parts of the independence movement in India. So this was the beginning of the end of colonial rule in India. Now, during this third plague pandemic, a lot of science was going on. And we've talked about some of this. In the 1860s, Louis Pasteur, if I can spell it right, was able to show that bacteria, well, he discovered bacteria, and he discovered that this was associated with disease. Now, bacteria had been associated with disease before, but the previous concept was that bacteria were something caused by disease. So we knew that bacteria and disease went hand in hand, but we thought it was the disease first. So Pasteur developed what we call sterile technique. He was able to use sterile technique to grow pure cultures of bacteria, which is really important for figuring out that bacteria cause the disease. So the way he did that was he made these, this is called a swan neck vessel. He would put growth material inside the vessel, so something that bacteria could grow on, the food for the bacteria. He would boil it and he would wait, and there would be no growth. So this sterilized the liquid. Just to make sure it wasn't something else going on, he did the same procedure, but then broke off the top part, the swan neck part, so that the vessel was open to the air. And sure enough, you get mixed growth of various different kinds of bacteria. So what's going on is the bacteria in this bottom image are literally falling into the flask. In the top image, this skinny, narrow, curved neck, bacteria could get into the opening, they could fall into the opening, but bacteria cannot travel long distances inside a narrow glass tube when there's no nutrients involved. There have to be nutrients involved. So with no food, they would starve to death before they could make it up this upslope and into the flask. Then they were able to grow pure strains. So they could grow just one type of bacteria. This open flask, you've got all kinds of stuff going on in there. And when you have a pure strain, you can inject it into healthy animals and see if they get the disease. And as it happens, you can. So Pasteur paved the way. And then Robert Koch developed what's now known as Koch's postulates. He learned how to grow bacteria in a petri dish. So he learned how to grow pure cultures of bacteria on a semi-solid membrane or semi-solid material. It's kind of a soft, squishy, but it's a food source for the bacteria. And he could stain those bacteria and look at them under the microscope and confirm that they were all identical. They could also take bacteria from this plate, inject it into a healthy animal, and see if that animal got sick. So the steps that go here, you get a sick animal and you isolate a bacteria. Nowadays, we can also isolate virus, but back then it was isolating a bacteria. Then you would grow the bacteria in pure culture, meaning it was the only bacteria growing in that culture. Then you would inject that bacteria into a healthy animal and observe if the healthy animal got sick. And then finally, you have to isolate the original bacteria from that second animal. And this was how Koch showed that a specific bacteria causes a specific disease. Now, there are some problems with Koch's postulates. I'm going to send you guys into your breakout rooms and see if you can come up with what kind of problems there might be with this type of experiment. Why might this not always show, or might not always be able to be used to prove that a specific bacteria or a specific virus causes a specific disease? So I'm going to send you into your breakout groups for 5 minutes, and then we will come back here and see what you guys came up with. Okay, I'm giving you 1 minute and I'm closing the rooms. Okay, what did you guys come up with as possible problems with Koch's postulates?
My group was saying that if there was a disease that was maybe non-transmissible through rats, or rats wouldn't necessarily be symptomatic to it, then a rat would appear healthy, but still have it in its system.
Yep. Yep, that's a good one. Anything else you guys came up with?
So I actually looked up some limitations to it, and they said that if a particular bacteria can't be grown in a pure like cultured lab, then like it would be hard to get results from that.
Yep, absolutely. That's another good one. Not all individuals are susceptible to all diseases. That's another problem. So there's quite a lot of problems with Koch's postulates, but it is a good starting place for understanding disease transmission, and more importantly, for demonstrating that bacteria cause disease. Back in the early days of germ theory. Okay. All right. So we're starting to get an understanding of germ theory, starting to understand how things are going on, how diseases get spread. My cat is very excited. There's a rabbit outside the window. During the third plague pandemic, the bacteria that causes plague was identified. And there were two researchers who were working really hard on finding the cause. And they were both working in China, because that's where the pandemic was more severe. It wasn't very widespread in Europe. It was more widespread in China and in India. So they were looking in China. We'll start on the left. Kitasato Shibasabaru, probably horrible pronunciation, I apologize, was a Japanese physician who had studied under Koch. So he had learned from Robert Koch all about Koch's postulates. And he went to Tokyo. Okay, so technically this was in Japan. And he was finding bacteria in plague victims that caused plague in mice. So he was already starting to find that there was something in the rodents that he could grow, that he could cause the disease in rodents, or he found the disease in people. Then there was Alexandre Yersin, and Yersin was a student of Pasteur. Remember, Pasteur was one of the early people who developed the technique to grow pure cultures. And Yersin was working in Hong Kong, and he also started finding that he could get bacteria from people who died of the plague and put it into rodents and cause disease in rodents. Now, there was competition between these two men. And because Yersin was European and Shibasabaru Buro, sorry, again, don't know how to pronounce that one. Since he was Asian, he reserved all the plague bodies in both Japan and in Hong Kong for himself, for his research. So Yersin had to bribe morgue attendants to get samples from victims of the plague. Kitsato, I can pronounce the first name better, found the bacteria first. He identified the bacteria and he published. In science, publishing is everything. If you don't publish your results are not going to be accepted by the scientific community. And publishing doesn't mean in the newspaper, it doesn't mean in like Time magazine or on a website or anything like that. It's in a scientific journal, and your paper gets reviewed by other scientists before publication. So publishing is a big deal. Unfortunately, for Kitsato, his description was not very good. His samples were contaminated. There were multiple strains of bacteria in there, and he had to retract his findings. He had to say, okay, I didn't isolate the correct bacteria. He was in a rush to get that publication out first, and it led to sloppy work. Yersin discovered the bacteria, and about a week later, he published his results, and the bacteria got named after him, Yersinia pestis. So, third plague pandemic ends in the 1960s. And let's look at the distribution of plague since the end of the third plague pandemic. So plague is endemic to lots of places around the world. It can be found in rodents in all of these places. So we can see the red areas. So there's the Western US, some in Mexico, Peru, Ecuador, Brazil, other spots in South America, various places in Africa, in Madagascar, in the Middle East, Mongolia, China, India, lots of places around the world. There are, on an average year, between 1,000 and 3,000 cases of plague in humans. Most of these are bubonic plague, and most of these are small localized outbreaks. In the US, in this century, we see between 1 and 17 cases per year. That's not very many. When I checked this data, I think it was probably 2 years ago when I updated it last. So between 2000 and 2021, there were only 12 deaths. So a very, very low case fatality rate compared to what it has been in the past. Madagascar had a pretty severe outbreak in 2017 with over 2,300 confirmed cases and about 2,000 deaths. So if you're doing the math, that case fatality rate is a lot lower than what we saw during the earlier pandemics. So the case fatality rate nowadays is about 15% for bubonic. It's about 40% for septicemic. Any septicemic infection, septicemia is when an infection gets in the bloodstream. All of them have relatively high mortality. Still nearly 100% for pneumonic. So pneumonic is still the worst form. Why has the case fatality rate gone down? One word: antibiotics. Plague is caused by a bacteria. We now have antibiotics. Antibiotics can kill off the plague bacteria. It can help people survive. All right. Now, antibiotics, supportive care, great, reducing mortality. Should we still be worried about the plague? Yes or no? Any opinions out there?
I would definitely say yes, just because, you know, it is devastating. You know, it has been proven to be devastating, you know, to populations. So there's like there still should be, you know, some fear, you know.
Mm-hmm. Yep, especially because of antibiotic resistance. So there's always the risk that your Yersinia pestis will become resistant to modern antibiotics. So yeah, I would be concerned. Okay. So that wraps up plague. One of the deadliest pandemics, the deadliest pandemic that the human race has ever seen was the Black Death. Far and away, it killed a higher percentage of the world population than anything else. Let's hope it doesn't come back like that. So I know I don't normally send you into breakout groups twice, but our next topic is on biowarfare and bioterrorism. And I want you guys to see, well, you're welcome to chime in now if you don't want to go into breakout groups. What do you guys think is biowarfare or bioterrorism?
Wouldn't biological warfare just be warfare that would take place on a biological level, like altering DNA? Well, not really altering DNA, but say something like nerve gas, neurotoxins, stuff like that.
That's a good guess, but that would actually be chemical warfare. Even though it's impacting people biologically, it's caused by chemicals. Anybody else want to try?
So isn't it like, so it's known as like germ warfare, which is like the use of like toxins to like bacteria, viruses, insects, or like plagues or anything,
Yeah,
to like kill like humans
yeah,
or like animals or plants as an act of war.
yeah.
Okay.
Yeah. That's what we're talking about here. Using usually infectious organisms, so usually bacteria or viruses, to attack your enemy or attack his food supply. So you might use a plant virus to wipe out his crops, or a disease that impacts livestock to wipe out his food supply. So this discussion of biowarfare, bioweapons, bioterrorism, it's kind of disturbing. I am leaving out a lot of the really horrific stuff that went on So, like, I'm not going to discuss the kinds of things they did during Nazi Germany, for example, because that's more than I can stomach. All right. So, defined by the CDC, this is what the CDC calls it: the intentional release of viruses, bacteria, or other germs that can sicken or kill people, livestock, or crops. So that is the CDC's definition of biowarfare or bioterrorism. Now, some people do include toxins if those toxins were produced by microorganisms. So you guys were close when you started talking about toxins. But it's just a subset of toxins. It's not necessarily things like nerve gas, which is a chemical, but it can be things like cyanide. Well, no, cyanide. Yeah, cyanide is a molecule, but it can be produced by organisms. Or botulism toxin. Botulism toxin is one of the deadliest toxins known to man, and it can certainly be used in this way. It's just hard to get it into people in large scale. It can be used to deliberately kill people, or it can be used to cause fear, causing terror in a community. The risk of using biological agents is that they can spread beyond their original targets. So if you were to release a biological weapon, say a modified antibiotic-resistant plague bacteria, there's no guarantee that that plague bacteria wouldn't come and infect you and your troops, your military, your civilians, your cult, whatever. So that can be, that can cause a lot of hesitation about using these sorts of things. When we look at the history of it, it is often very difficult to tell if an outbreak was caused by natural causes or if it was some form of bioweapon. We would like to think that we could look back at history and say, oh, yes, this happened at this time. But the truth is, the winners write the history books. And what actually happened is often misrepresented for political reasons, especially if the winners did something horrific, like use bioweapons. They might not want to write that down. They might want to wipe that from the history books so that it doesn't come back and, make them unpopular or cause their own population to rise up and kick them out of power. The passage of time always distorts things. We try and record things, what's going on, what's happening, but over time, no matter how we record events, whatever we recorded them on is going to decay. Paper, chiseled stone, computer disks, all of it eventually will break down, and terminology changes. So I mentioned the other day that the phrase 40 days and 40 nights originally meant for a very long time, we don't know how long. But it has been interpreted as exactly 40 days and 40 nights. So that's the kind of thing that I mean, that we can lose the context. And misunderstand things that have happened in the past. So let's talk about documented cases. The first documented case that we have written evidence about is from 1400 BCE, before the Common Era, formerly known as BC. And there was a group called the Hittites. And is that spelled right? Hittites. That I should not be there. And the Hittites sent infected sheep to mingle with their enemies' flocks. So they would get sheep that were sick and send them into the flocks of their enemies. We think the disease that was really happening was a disease called tularemia. Tularemia can infect people. It can infect sheep, obviously. It can infect a wide range of different animals. In sheep, it causes fever, weakness, abscesses, which are swollen, pus-filled pockets under the skin, pockets of infection and immune system cells. It can cause miscarriages. Death would occur often in 1 to 2 weeks, especially in the very young. Tularemia naturally can be found in rabbits. So the rabbits seem to be a reservoir for that particular disease. This is an example of somebody who's infected with tularemia. So you can see this open sore on the thumb. This is pretty characteristic of this particular disease. So this was often the way biowarfare happened, especially before we understood germ theory. We understood that diseases were contagious, even though we didn't know what caused them. We would also see archers that would dip arrows in decomposing animals, hoping to pick up the disease. I'm sure sometimes it worked, sometimes it didn't. We would see especially the use of things like venomous snakes. So venomous snakes, we know they kill people, so we're going to kill the snake, dip our arrow in the body, hoping to pick up whatever it was about the snake that caused people to die. Obviously, that's not going to work so well. The venom's in the bite. They would use the saliva of rabid dogs. Rabies, in case you don't know, is pretty much 100% fatal. You don't want to get that one. They would use extracts of poisonous plants. That was probably more successful than the snake. We talked about catapulting or throwing cadavers over the wall. We know this happened during the early part of the second plague pandemic. The most recent case of this, literally catapulting bodies over a wall, happened in 1710. And the Russians laid siege to the Swedish army in an area that is now part of Estonia. So Eastern Europe. People would dump the cadavers in wells, poisoning the water. Certainly, I don't want to be drinking cadaver water. Even if I'm not going to catch a disease, that's some pretty nasty stuff. Selling or giving potentially contaminated goods to the enemy. We saw this in the Americas, where British officers would give blankets from smallpox victims to the Native Americans. We don't think that was very effective, but it may have killed a few people. Confederate, this is the Civil War in the US, the Southern Confederate public would sell clothing to invading Union soldiers. And they would sell clothing from smallpox victims and yellow fever patients. Yellow fever is a disease we have not discussed. It's not on our agenda, but it's another pretty deadly disease. To really gross us out, the Spanish would sell wine to the French. They were at war with the French, but they would sell wine to the French anyway. Got to make money somehow. And they would add the blood of leprosy patients to the wine. Again, the gross out factor there is really high. So there are lots of examples of trying to use biological weapons in various ways to spread disease among the enemy. Nowadays, we can categorize biological weapons into three major categories. Category A is the most dangerous diseases. These are the diseases that this is from the CDC. So these are what the CDC worries about most. The World Health Organization has a very, very similar list. So category A, these are diseases that have a high mortality, that spread easy, and that would need to be targeted and smacked down quickly if they start showing up. So this can include Bacillus anthracis, which is anthrax. Anthrax has been used a lot. It's a very easy bacteria to find, isolate, and spread. And it was used in World War I, World War II, the Soviet Union in the 70s, Japan in the 90s, and the US in 2001. We're going to talk about those cases, or the cases in the US at least. Botulism toxin and Ebola, those are both highly deadly. I didn't find any examples of those ever having been used. There's a disease related to Ebola called Marburg. There's a small outbreak of Marburg happening on the continent of Africa right now. I forget exactly which countries are impacted, but it's a hemorrhagic fever. Hemorrhagic means it makes you bleed. So hemorrhagic diseases, they'll have you bleeding through your mucous membranes, internal bleeding, really, really unpleasant. We know that the Soviet Union was trying to figure out how to weaponize Marburg. We don't know if Russia is continuing that work. Plague, we know it was used in the past. We've talked about that. It was also used in World War II. We talked about smallpox in North America, variola major, and we just talked about tularemia. Tularemia was also used in World War II. So I hope you're getting the idea that a lot of experimenting with bioweapons took place in World War II. Category B, we call these public health hazards. So not as deadly as category A, not as easy to spread as category A. So these are still dangerous conditions, but they would be easier to manage and easier to treat. So there's a lot of diseases in this category, things you've probably never even heard of: brucellosis, psittacosis, Q fever. Probably, like I said, haven't heard of those, never been used, never been tried. Vibrio cholerae, which is cholera. Cholera typhus, another disease we haven't really talked about, and several other bacteria have been used in World War II. Salmonella and other forms of food poisoning like Escherichia coli 0157H7. So it's a specific subtype of E. coli that is deadlier than standard E. coli. We all have it in our guts. It's not that big of a deal. It's not dangerous, but this 0157H7 strain does cause food poisoning. And those were used again, World War II, also in the US in the 1990s. And that's going to be our other big story about, that's bioterrorism. Category C, these are newer pathogens or genetically modified pathogens. There's not a lot known about these. They might be easily produced. They might have high mortality, but we don't know a lot about them. They're kind of like let's keep an eye on these. These might turn into a problem. So we're not going to talk about very much of the stuff that happened during World War II. A lot of the things that I was mentioning that were going on during World War II have a lot to do with, frankly, the Nazi Germany party. They used a lot of chemical weapons. They used a lot of biological weapons, sometimes on the enemy, sometimes on people in concentration camps, just to see what would happen. Truly evil. So we're also not going to talk, we are going to talk a little bit about some stuff that went on in Japan during the Second Sino-Japanese War. So this was between China and Japan. It happened about the same time as World War II. So if you recall from history, there was the US was involved in World War II in Europe, but they were also involved in the war in Asia. So we were working both the Atlantic and the Pacific Ocean at the same time. So these two wars often get lumped together as World War II, but they had different origins, whether we're talking about Europe or Asia. So I'm going to start off our biowarfare with Unit 731, also known as the Army Epidemic Prevention Research Laboratory. This was a Japanese program that started in the 1930s. And despite the name saying epidemic prevention research, the goal of Unit 731 was to develop biological weapons. So they were looking at how to produce large amounts of viruses and bacteria, how to deploy large amounts of viruses and bacteria, or how to spread it among the enemy, and storage and transmission, right? So depending on the source, here we go again. History gets rewritten. The details of what actually happened during Unit 731 vary dramatically. So I'm doing the best I can to pull from these different sources. They did use human subjects, so they weren't, well, they may have been experimenting on animals, but they were experimenting on people, predominantly prisoners, homeless, mentally ill and captured Chinese. Because remember, China and Japan were enemies at the time period. They would infect them and study the course of disease. We know at least 10,000 Chinese were killed during this process. Probably the total numbers who died during this time period was much higher than that. They tried to develop bombs that could spread pathogens. They tried to develop methods of infecting water supplies, wells, reservoirs. This is something we see over and over and over. We talked about dumping cadavers in wells. The Japanese were trying to do a more modern version of that. During the Second Sino-Japanese War, which was 1937 to 1945, same time as World War II in Europe, Japan would drop plague-infected fleas, infected food, and clothing, again, laced with pathogens, into China, deliberately trying to spread these diseases through China. It is estimated that their biowarfare efforts caused the death of between 200,000 and 500,000 people. It's a broad range. We don't have a lot of good information. The survivors, the winners, rewrite the history books, but we know that this was relatively successful. As far as I can tell, this was the, excluding whatever the Nazis were doing, this was, okay, if we include the Nazis, this was probably the second deadliest use of biological weapons in history. Most of the time, biological weapons didn't really have a significant impact. In this instance, they did. Japan was also accused of using biological weapons against Mongolia and against the Soviet Union, what is now Russia. They had plans to send the plague to San Diego and other places in the mainland US, but Japan surrendered and brought World War II to an end 5 weeks before the attack on San Diego was scheduled. Just barely stopped this sort of thing from happening to the US. Members of Unit 731 were captured by the United States. What do you think the United States did with them? You think we were the good guys? Put them in prison? No. They were given immunity from persecution for war crimes in exchange for their data. So the American government wanted the data from this program more than they wanted to punish the people who did this. I'm going to leave you on that horrific note. Oh, I see comments in the chat. Okay, so that wasn't for me. You guys having trouble hearing me, huh? I'm sorry about that, but it's time to wrap up. We will come back to this topic next time, and we will talk about what the US was doing, and we will talk about bioterrorism in the United States. Have a good weekend.
Have a good weekend.
Thank
Thank
you.
you very much.